Let's study together with metabolomics, with LCMS or GCMS to analyze. Yesterday we already tell you about the difference about GCMS and LCMS, analyze the metabolite, and they have different the targets or the emphasis on the metabolite. So I will try to use sometimes cross-law or what. Sometimes I remember, okay, this point is very important, but I will do very detailed, like this. Okay, what is the GCMS? What's the difference, like carrier gas or mobile phase? Those kind of things. I guess I hope yesterday you can understand some of those differences. And actually, all the GCMS. Let me simplify the GCMS or LCMS, the difference. One is for volatile or for volatile compound, or for some metabolite can vaporize into the gas, so we can analyze it. We can be ionized to analyze the mass spectrometry. For liquid chromatography, mostly they use some larger molecule, very difficult to evaporation. So in that way, we have to use the LCMS. But actually, in this world, the most substance is cannot be evaporated. So that's why LCMS actually used much more, and also there are some larger molecules cannot be evaporated. Usually can use have to be used the LCMS, and also use the other, like we studied by organisms, what different lifestyle. So we have to heavily rely on the LCMS. So this part. But anyway, the first you have separation, GC, and LC, UTLC, or HPLC separation part. Then the separate solute or the solute gas carrier, they come into the mass spectrometry. You get in GCMS, there are vacuum to analyze those ionized metabolite. For LCMS, they are not vacuum, but they have very different mass spectrometry, like a Q12, and here we mentioned our Orbitrap, our the tripod, the Codal. So those different tools are analyze, mass spectra really increases the powerness of this mass spectra. We can analyze different molecule weight from 100, 200 to even 2,000, or even like 10,000, much larger molecule. So the power actually dramatically increased, like in the Orbitrap. The Orbitrap, that is a very high sensitivity and also have a very high capability to analyze the larger molecules. So for that, another interface is before getting into the mass spectrometry, the ionization or method is different. ESA or electron ionization, which normally used in GCMS, ESA, or other chemical ionization, or APCI, all those different ways to do the different target of a metabolite. This really depends on your interest. And this different type of mode we also mentioned. And later on in lipid analysis, I will see the more we use the selected ion monitoration, because larger molecule you have to select ion peaks. We also tell you about the principle why those ions can be, why those particles or substance our sample can be ionized, and during ionization, the ion of the molecule or proton change can make the charged particles or metabolites unstable. Those unstable very easily fragmented into the different even neutral molecule or small ions. Those can be analyzed in the detector. So this way you can get the different type of the fragmentation. So actually from those tabular mass, our triple quadruplet, our Q-trap, those different ways, Q-trap we also later analyze, distinguish those from the Q-top or triple quadruplet. Those really use a different method to achieve the final goals, is to distinguish the different molecule in separation part or in ionization, and before be perceived by the ion detector. So there is the time of flying, travel time, differentiation with the different molecule, and also orange trap with OB trap. Those metabolite ions, the turn around orbit with the metal spindle, then the different molecule, and also their charged ratio cause different stimulate, and finally in the ion detector use a different signal. But those, if you get I also mentioned the OB trap and the cold open difference in why they couldn't. Why OB trap with the high sensitivity can analyze larger molecule, but they're not very suitable for metabolite profiling for one time or through the long channel electric field to get the metabolite detected in one time. So this is very different. That's why the Q-top is much more optimal for analyze to do the metabolomics analysis. So this is one major point. There are some fraction, actually it's not very okay. I couldn't see very clearly about this PPT, but anyway, there's some difference about this use the LCMS to analyze the blood sample. You can see the one example, one is the application about the utilization of the LCMS, and also another one is analyze the phloem sample, which is the clinical phloem sample. You can do that. This is the LCMS. And, I think this LCMS to find the different sugar or amino acid or other phosphatic metabolites. Finally, you can use those metabolites to use the KGT or very common the pathway analysis to put all the metabolite what you have analyzed into the pathway. Yeah, that's later on we also discussion. We will also mention and also today. Dr. Wiener actually already mentioned this. This use GCMS to analyze derivatization the sample. There's many samples if you want to apply to the GCMS, which have many different. I think they have very good advantages for analyze. They have a very good library for mesh. You don't really spend a lot of time on the ion peak identification one by one. So much more convenient. So all of those you can see there are ME methylate, the methylate ester, the methyl ester. All use the methyl ester. Just this is just one step of sample preparing. First, extract lipid, and then use after lipid dry, then we dissolve into methanol, and then add the alkaline, like hydroxy potassium or hydroxy sodium. Yeah, under the very high the alkaline condition, those will be methylated, just like we prepare the sap, that they also call the stabilization and amino acids. You know, we use the animal fat together with the alkaline. Superficial indication. Yeah, similar. The mechanism is the same. As long as the larger molecule of carbon hydrate was methylated, they will be very easily evaporated into gas, then easily analyzed use GCMS. The trends of their appearance about the time over the analyzing time, you can see the high molecule you see the show up later. So that's exactly you can tell, okay, the molecular weight to the charge ratio. Charge ratio really depends on those molecule weight and charge are basically very similar. So yeah, this is very important. And also GCMS shows that actually it's the 3D variant or even, sometimes you can see 2D variant. But the powerful you can see is the extraction time over running time and also energy and also intensity. You can see the difference, high resolution. So that's why I prefer the GCMS sometimes use the very normal. They have the 70 electrowatt EI, the library. You're pretty. EI is the electroionization. That's what typically used by GCMS. This is some interface about the GCMS analysis. The own library you can directly use. Usually you buy GC, you have a software installed inside. Yeah, then just use that to analyze the metabolites. They will give some protocol for different type of either amino acid or some fatty acid. All those need to do pre-treatment, derivatization. So this is another interface. Later on, when we do the data analysis, we will also see those kinds. But those largely for the LCMS, because LCMS, the application, a range is much wider than the GCMS. Yeah, it's because probably many analytics, chemical analytics spend much more time on the LCMS, the power to increase those. This is 2D variant, and this is 3D variant. 3D variant. Just focus on the separation. Separation part is very, very important for the GCMS. You have good separation, you definitely have much better the molecular metabolite identification accuracy. This is one. Yeah. So this is.
Last day you completed this.
Yeah, the TIC is very important. So this we will see. Yeah, this is one example, right? Yeah, we see exactly structure. How do you understand structure? Is the different after the larger metabolites was broken down, the one by one broken down. You will see the molecular ladder increase. The difference you can calculate, actually just minus or decrease. So example for the, this is for the protocol, the different, the QQQ, the first, the MS, and the second MS, and third MS have different function. You clearly between the two first and second, third MS, their collision cell fragmentation process. This machine actually can be used for the larger molecule like lipid analysis. this gluten, as is already trapped. The principle is shown here is for the peptide analysis is different. I think last time limitation for GCMS and some. We mentioned about lipidomics, which I have been working on. So just use those what the molecule that media to tell you how a large molecule was metabolite was injected into MS in which position you leave it broken down, then generate the typical or representative the daughter ion peak, and also use that daughter or use those the product ion, some also call it product ion, to represent the precursor ion. The precursor ion is the metabolite you are interested in. So I guess those you already know that we use the different molecular calculation. You find out doesn't matter you positively charged or negatively charged, you will see those different peaks. Just use the minus, usually minus the precursor ions, lose the one part and lose another part, generate different ion peaks. Those signal, if consistently show, that definitely 100% you identify this peak. The precursor peak is the what kind of metabolite. And this also just use ammonium, the positively charged metabolite mixed with the lipid sample, mixed together, then load up to the quadruple, triple quadruple, the ESI, LCMS. Find this peak. So, for example, I just use the details we can tell it more clear. For example, this LCMS fragmentation pattern of the molecular M is the precursor, the metabolite of precursor ion plus the ammonium. We use the ammonium to treat the sample, lipid sample mixed together. Then log on the machine, you will see the MGDT ion peak is 76.2. Is 762. So this the generated this major peak later, and then 582, 585 or another one. I couldn't see very clear. But how can you judge those? You can see if 764 minus 179. And from the combined loss of this one and also 162, those molecules is defined like a galactic molecule, because this is galactic lipid. The galactic molecule is 162. The plus 1 ammonium, the 17, then put together is 179. Then if after minus, you will get 567. This is the diacylglycerol, the structure. After the loss, you will get that. Then if you lose another one, you can get that. Get the third, the peak. That's how they calculate this through that. But this PPT, anyway, you will see that. So you will print it if you use it, so you can calculate and study those. And this one also is the SQTP, is a galactolipid contain a sulfur. You can see it there. Have a sulfur. This pointer this one is shut down. Oh yeah. Okay. Can you see that? A pointer to the skin on wall is okay, but the skin. Anyway, have sulfur, 3 oxygen sulfur acid. Attached to the sulfur galactose acid lipid. It's very important in photosynthesis. Okay. You also can calculate those with a certain we fix the fatty acid chain. The two fixed fatty acid chain basically besides the molecule is 575. But there, if you have a different other chain, the molecule change increase to 800 or it's all type of molecule switches. This lipid is very complicated because they have different two variation in the two types of variation in the two fatty acid chains. And of course, the polar type is the sulfur galactose part, moiety is fixed. This is the polar part. For other fatty acids, for other also like those is lysyl PC and PC, and also sphingolipid, SM, sphingolipid, and TAG, the neutral lipid. You also can do with this kind of the machine. You can see the structure attached here. Those PPT, I think later you can study. I think they will later print out to everybody that attachment. So you can see, and also there's some the publication. But the way we identify those peak or identify the metabolite is what? The mother metabolite or precursor metabolite is what type of molecule. But also later I will also show that doesn't matter lysyl PC or SM. You all see, as long as they have coin, they have one representative marker, the ion peak. Usually those hand, the lipid, I think initially we tell you the lipid have three, because they have a glycerol. Yeah, they have glycerol backbone, right? So three hydroxy can be estered with three types of fatty acid chain. The fatty acid chain change, like 18-1 or 18-2, 16-1, common 16-1, or even 12. Those will alter the molecular weight. However, the hand part is the choline or serine. The PI is for serine. Our PC is for choline. Our SLME, PE, all those in our body is very common. They have a large amounts of because cell membrane is consumed by the polar lipid. So the polar part actually can be a marker, ion peak. As long as you find those in this system, in this established lipid metabolite profiling system, you definitely see this should be PS. If this is lysol is just only have one fatty acid chain. Yeah, but normally the PC have two fatty acid chains, so this is kind of the difference. Yeah, all those PIP, MGDG, DGDG or DGDG means have di galactose, the chain in the head part. So the presence of all those molecules can be identified the presence of a specific head group fragment. Yeah, this repeat. The mass generally allows us to assign specific number of acyl chain. Yeah, if a head part is defined, this should be choline, or this should be serine, or SLME, or even inositol. As long as this is defined, then you try to define the acyl chain, acyl carbon part. The specific number or number of double bond. According to double bond, you will know the saturation degree of acid chain. Now also correction is applied to the data used for the isotope overlapping. That's why when we profile the hormone, use the Q-top MS, we usually use the isotope labeled hormone to do that. Many companies provided the standard, the isotope labeled standard for people to do the established method, and also many often we need to apply to adjust our system is working or not. Because every LCMS or HPLC, commonly like HPLC, or you couple with the mass spectrometry, you need to adjust the separation part and also the MS part, mass spectrum part. So adjustment usually need to take some time, you need to clean up, and then you readjust, use the isotope overlapping or other more accurate internal standard. Internal standard is much more less cost effective, but you need to apply the all. That's in later data analysis will also say you need to do the data alignment with the standard to get the correct peak. But we cannot find all standard, many unknown peak. Actually, we couldn't. So as long as our standard is scattered in our range of the mass spectral range, usually those match is very good, then will be great. Yeah, can good for next step analysis. So those are for the hand group analysis. And this is some data identification of most abundant signal in ESA mass spectra of a total lipid extract of animal cell. Yeah, the CHO is the animal cell there. But this, of course, sphingomycin only, the mainly present in animal cells. In human beings or in other animal cells, the sphingolipid is always related to a different kind of disease, like brain or other disease. So in plant cell, very few. The cell type is different. And this also shows the scan mode is a single stage negative ion or positive ion. Negative ion, we know we use the sodium or I think we use the potassium. The positive ion we use ammonia. And this is this is for the fatty acid fragment analysis, another kind of analyze. I just want to tell you how, still use the same way. When here is the precursor molecule, this is in negative mode. Yeah, you need the -1 to positive. Yeah, in that case, you have one over polar head loss, you get 70/1. If the molecular calculation is very well, then basically you can identify particularly the head part. The head part is the signal. If match well, that will be great. So. Yeah, this is another scanning for the ESA pattern analysis. So this publication is also, I think, in PAS, in PNAS to establish the method using for analyze the phospholipid. Because the recent year, the phospholipid study is really hot because those are not only related to our plant, the human nutrition, plant food for human nutrition, but also related to some stress. Every, the cell stress, the first attached part is the plasma membrane, right? Yes, every cell is surrounded by the plasma membrane. So those membranes have phospholipid. So those are also different type of this is a kind of table shows on the different collision offset. The collision part is what we said, Q2, right? The Q2 is to broken down the cell. Then we have broken down the particle. Try to broken down the particle as completely as possible. Then go to the surface mass spectra then go to the ion detector to receive the signal, then go to software to analyze those. So we can see the hand part is twofold. 1 or 87, 160, all those precursor, the typical precursor for the head part, right? Sphingomycin or sphingomycin, the head part, the head group, corresponding molecule, like this is phosphatidylserine, phosphatidylethanolamine, and sphingomycin, choline, sphingomycin, and also others. Because of the reflection, I couldn't see very well. So that's the detection of phospholytic. Also in different way to detect it. This in positive mode. In positive mode, and previous slides is in negative mode. In both modes, you can identify those lipid very well. That means the method actually is very powerful, very universal. You can do this is the tablet method. The previous my supervisor in Kansas State University, they use triple control, triple control. This is photo shows that internal standard of with over the molecular to charge ratio, you can see the different PC molecule spaces. The molecule increase and then signal decrease. Signal decrease. That's just because the molecule increase. Then this is the PC for another part of the internal standard of PC 82. 38.2 means you have two double bond, and total carbon number is 38. 38, it can be different combination of 16. It depends on what type of organism resource, like plant or animal difference. The 38 can be 18 plus 16 fatty acid. This is 34, right? 34 then plus 38. 38 probably have other PC. Also have polio. Then also 34. 34. I don't know. This is for because this animal cell probably completely. Usually we can use animal. In plant cell, I can say 34 is very common. 36. 34, 36, like a double 18. But anyway, use those. You can do the quantity. You can do the quality connecting identification of which molecule or which metabolite we are detecting, and also we can do the quantification. The quantification, I think the one over. Quantification. The good thing is that they do the metabolite profiling. So many complicated PC have different molecule species. I think PC have more than 30. More than 30 is different, depending on the organism, the molecule. So in that case, how do you quickly do the identification? So this kind of job is very significant, just because the very complicated the molecule species. Only PC you have so many different variety in molecular structure, and also no matter the PS or PE or PI or other. So that's why they developed the software, can regularly check this. As long as they fit their templates, they can automatically output all those metabolites in this Excel table. So you can quickly use their template. So if you ask them to analyze those data, not only reliability is high, but also the easy simplicity is much higher. So that's why their save their favor, save their labor and time, but also give you the very high reliable, accurate data. So they set up the template to large scale, in large scale to analyze those data into the result. So then you can do the figure out, do heat map, then do comparison. For example, this is a biological sample and with another biological sample. What's the difference? Is the difference in the PC or PE? Because this we already examined those accuracy in some Arabidopsis mutant. In Arabidopsis mutant, if we deplete our mutants are phospholipid, those phospholipids specifically hydrolyze the PC in membrane. They attach to membrane, hydrolyze the PC of our PE. Of course, they're not very specific. Sometimes they can hydrolyze the PC, but they also can hydrolyze the PE. But they have preference. The reason we know they have preference not only because we express this phospholipids in cell, in E. coli, in prokaryotic cells, but also in that cell after expression, then we purify the enzyme, then we bind the standard, then do the enzyme activity assay. We can see the head part was hydrolyzed by these phospholipids, because this phospholipids just remove the head part and generate the piece, generate the phosphatidic acid. The hydroxy part was removed, the choline was removed, or SLM removed, generated the phosphatidic acid. From those mutants, we can detect dramatically decrease or dramatically increase the PC when we mutate the gene, or when we increase the gene expression, we can see dramatically decrease the PC. Because increase the enzyme expression, you can see the plant phenotype change, and also PC, the content is dramatically reduced. The reduced PC sometimes corresponds to the increase with other phospholipids, like the PE increase, to complement the defects. So they can sometimes replaceable, can back up some part. So that's why we can see this, the difference in both the overexpression plant, in mutant plant or knockout plant. You can see the corresponding change of the enzyme product or substrate, the change. So this naturally should be very standard. The ratio of PCPE normally is very consistent. However, after mutation, the certain type of gene, you can find, for example, phospholipase D, phospholipase C, and phospholipase other type of phospholipase A, and those fatty acids. So those data actually, by comparison, we can interpret the biological meaning of not only the gene function, but also under different environmental situation, for example, attached by the pathogen. Yeah, they also change the membrane components, that's for sure. And also under different abiotic stress, drought stress, cold stress, and the membrane fatty acid, membrane lipid change dramatically. So, yeah, this help us to identify. Anyway, this compared again, this is why I came back. Okay, this so can we? Yeah, I think I will change it. To interpret the MS data. Yeah, if you have any question, we can we can discuss now. Use the GCMS to analyze long chain carbon hydrogens, like actually this fatty acid. Just because this fatty acid, they have a COOH, then we also have a methanol, acid and ethanol, or alcohol, methanol, alcohol, ethanol, alcohol. The alcohol combined together will become ester. Yeah, lost one water. Yeah. Yes. H2CO, lost one water, then generate the ester. The ester will be very easy to be evaporated. Yeah, to be evaporated. Then facilitate the analysis by the GCMS. So we use that example to show that in GCMS also similar principle to analyze the mass spectral ion after in the electric field under high energy, like usually 70 electron volts, they will generate those kind of the fragment. They will be broken down, unstable broken down, and generate the fragment. The fragmentation, if you use the tandem mass, you can see the continually detect M1. In the result, in the data, the machine running, we don't know just the one box. But in the data, we can show you MS1, MS2. Yeah, this tandem mass, two mass connect together. The triple mass is in middle. They further use the high energy form a collision cell, further broken down. Yeah, this is very important. The reason we have a low identification or low sensitivity just because some iron, after all, they don't broken down. They don't have a certain iron peak. You fragment the iron peak. So we could be sure which is still larger, it's the same molecule, or only when you broke it down, because each chemical structure under the high energy electric field, the iron charges, they broken down have certain rules. For example, phospholipid first broken down at the head part also. In head part, the first broken down. Then in fatty acid part. So then you can see the different fatty acid group remove this like 18-1, get the one ion signal, I get another signal, I get another signal. But some parts just can only get one. But those head signal already give you exactly what type of phospholipid. If it is phospholipid C or phosphatidyl choline or phosphatidyl inositol or those. Yeah, that's why they also use the selection, the filtration. That's why they are analyzing the multi-selective ion mode. Just because they want to select, they want to, when you show up the total ion TIC, yeah, like we always show. In this PLC, their total ion peak. But if you enter into number, for example, I enter into 182, 184, they probably give us, then we enter the 184, we only to look for molecular and ion and charge ratio with 184. 184 you will see probably several, but sometimes you only see one. If this is lipid, that could be the choline. Then this total ion you need to do the selection, then you do the peak identification. So I think for those I have any question about how to analyze, because those is real technique.
So we can take it after the lunch, because if you take it fast, no, we could not understand.
Okay, okay. If you have any question, yeah, please let me know. We can discuss why we, why usually MS, LGCMS, or LCMS works. You can identify different iron peaks. Actually, just use a similar, very simple plus, minus, and give minus. This really depends on the machine. Even sometimes, yeah, just like Dr. Wiener also told, later on we will have a lot of artificial intellectual, those software or technology involved in those identification. Yeah, the Dr. Wiener shows so many websites to analyze the data, and this here also will emphasize some. But you will see very clear trend that in the future, analysis of the metabolite profiling will be easier and easier, much more easier.
Professor, have you given your slide to Vinay sir for?
Yeah, sure. Yeah, all those.
Because ना before
you should
नहीं
be
sir,
there.
अभी जैसे जब लेक्चर लेते हैं ना, तो हमारे सामने अगर वो स्लाइड्स रहे ना, तो हमें काफी इजी जाएगा सर, बच्चों को भी इजी जाएगा.
Sure, you will share all my slides, yeah, so that we
No,
can.
before, no, during your lecture we need your slides.
Our
1 hour.
Yeah,
So
sure.
1 hour. Exactly.
1 hour?
Huh?
It's 1 hour. How much one? We need the time to cover these.
Oh, we are probably need more time.
More time.
Yeah.
Then we have to declare. And then after lunch, sir will explain his work. Then I will explain my another things actually associated with this.
Okay.
So it was only this recording is going on. No issue. We can start.
Yeah. If you have anything in the hand space of your sample, for example, you put some leaf, you put it into the vial, close it, then put a fiber in it. In certain time saturation, the fiber will absorb the gas. The oils, fragrance, or evaporated, the monoterpene, those fatty acid, oxidation products, or the oxidating, different type of carbon 6, carbon 9, carbon 12 to carbon 14, like jasmonic acid, also can get together. Then you use the saturated the fiber to put in the LCMS. After heat, then we'll dissolve it, then get into the carrier gas for the LCMS analysis.
So
This is very
directly you can take the sample and put it into the analyzer.
Yes, the sample when you put it into a wire, glass wire, then need to be sealed with a rubber
और उसी
sealer.
सीरीज में से लेने का
Yeah,
और उसको
then there's a
injector
small
में
hole
डालने
there,
का.
you put your fiber there. The fiber is produced by something like a sigma. Yeah, sigma has it. The fiber can be reused, can be regenerated, so the heat or some method. So can reuse it for a couple times. So even expensive, but you can reuse it for sometimes if you care very well.
What exactly is the name of the fiber? Because if I want to see in a
Silicon fiber. Yeah.
Silica fiber.
Yeah, they also have other type of fiber. It depends on your analysis.
Silica fiber.
Yeah, those fiber you can see is inserted
Yeah.
into
This
the
is the
wire.
fiber coating.
The vial can be heat. You can heat the vial, the sample. For example, you can heat, for example, our tea or tea grinder. We just smell the tea. Dry tea powder will still very good smell. So what's the smell? The components, what's chemical composition of the smell? So we can analyze, just put the power into the vial. Then we can put into the water bath. Or you can put the saturated salt, the sodium chloride. They already show that initially we just put water into the mix the water, the powder dissolved in the water. But finally found that the water can catch many water. But if you use saturated salt, sodium chloride, you will get much more better release of fragrance. All those.
It means it takes out water from the solution, no? Fiber.
No, not
The
less.
sodium chloride, which you are using as a saturated.
Yeah, this, no, the only solution is inside the solution you have the leaf tissue or something. You try to let them release more. Right? That's why sometimes we heat. But if you try to release more, sometimes you want to grind. You want to grind those into powder. Then you dissolve in a solution. The solution in water is not very good because the water can hold on many metabolites.
Okay.
However, if you put dissolve in the saturated sodium chloride, the sodium,
Do you need to heat them or
If
you
required.
can?
yeah, you still can heat.
Okay.
The slight heat, you will make the head space free of those molecules. Then the fiber will absorb more. Yeah. Probably for 2 hours, 1 hour, they will keep the saturation. They will reach saturation. The saturated the molecule, then you will take out and put into GCMS. Yeah. So this method I have to tell you is very important.
This is really very important, and you can understand that this even a single LCMS require 10 days of the training to understand the whole concept of the LCMS. Yes, it has so many variations, starting from the chromatography up to the ion detections and then acquisition of the data and finally analysis of the data. But yes, this is something different. Here you can use the sample. So many sample has some secondary metabolites when these infect and the pest come on the plant. So they release some terpenes and some other components also in the surrounding area. You can smell those plant leaf tissues also. You will identify a unique smell is coming from a particular plant tissue. So if you trap those components and analyze during the infection stage and during the normal stage, so you can compare that what terpenes are released and what is the function of those secondary metabolites, terpenes, with respect to that infection. So for that type of the aromatic components which are released in the environment, for that this is the best accurate chemistry. So thank you, Professor Jack. We will meet again after the lunch, and now there is a break for the lunch. Thanks so much. For the white, but let's see, Ritu is coming. If she can do it, it's okay, otherwise we have to wait for the 5 minutes. But he is going to start now because we have to free on the time, and just that is the better knowledge. So now really a scientist is known by his research work and what the techniques he used actually. So to understand the basics, to teach the subject in a class is a different thing, and to do the research is a different thing. As I know about the work of Sir, he is doing really fantastic work, and recently Sir told me that he submitted a paper in the PNS, and his target is to go beyond that, the PNS. So yes, he will explain all the data sets and whatever the data. It was my class, but I shifted him because whatever the time he will take, it's okay. Rest of the time I will manage my class. So I would like to invite you. You just wait to start, and someone come and just rectify it. Loudly, please.
बस 11 मेरी हमारी अभी 2 और आएंगे इस प्रोजेक्ट में सैलरी ट्रेनिंग हमारे सारे बच्चे
The problem is that right now the PPT here is not synchronized with this one. So I couldn't see what I'm talking about. So it should be synchronized, for instance. So I can see what the screen shows, so I can follow that. However, so I have to try to see, when you see here, the flash and...
We are able to see, sir.
Huh?
We are able to see.
Yeah, you can see, but I couldn't see.
Yeah, that's
So I'm working in this paper here. This snatch. You can see, oh no. so that I can see what kind of protein we are in same page. Otherwise, we always use kind of some PPT use kind of a mass, and some is protein. If some familiar, yeah, we call it MS. But anyway, the mechanism is similar. Use the kind of mass, I think that one have to use the very most recent renewed, and some use the triple quadruped. At the middle, you have a collision cell. The collision cell increase the fragmentation of this metabolite. So increase the sensitivity and also increase the capability of identifying those iron fragments. And eventually we can track back to find what is the precursor iron, what is the metabolite that we have already loaded in the sample, in our biological sample. So this, we show that we need to find out what's the rule of those lipid molecules that are broken down. So I showed you that the first we need we can see the PPT change. Actually, that is my habit. My teaching experience tell me that when we talk about something, we have to briefly review the previous slides or previous talk, so you can recall your memory. So we can keep in the same piece, put it in the same page to together study or learn those scientific information. So that's why every time I usually go through previous several PPT to show the previous what we have told you. For example, this morning I told you yesterday we talked about the top and talk about the triple quadruple, the MS. Those are the major part. Ionization is when we bind the thing, we do selection. We know our target. We know this capability is higher. We already can cover our metabolite profiling purposes. So we buy that machine. During by this machine, the ionization style already selected. There's no way to change. The machine fixes it. So whatever those ionization, the download the mass spectrum is very important. Of our triple photon, our orbitrap, those different style. Most recently used very widely in all type of industry, or academic, or agriculture, or the various kind of purposes. For example, for the agrochemical analysis to monitor the export of the food, our agricultural products not contaminated or whatever other safety issue, or in health study, in biomedical study, we need to analyze biological sample to make sure those metabolites we know, and we also can find out them. So this application is tremendous. So we also use that to measure some, use the lipid as an example. But later on, probably tomorrow we will have break, and for next Monday I will also give you some data. This just use myself data to show that flavonoid, the most common metabolite, secondary metabolite many people study, how those metabolites in our regular LCMS, the pica ion shows how their rules. For example, we know there are several ion peaks like 274 or 180 or something. Typical iron peaks that show that this is S-thymine. Those are catechins or pyrocyanidium. They from similar pathway, then separate, branch out to its end products, S-thymine or pyrocyanidium. But there are flavanol, the earlier branch out, flavanol or flavin. A flavin, those like arginine, or luteolin, or other kind of, and also isoflavone, those branch out the stuff pathway. It actually all shows different characters. Those samples will go through the LCMS, they will give the different ion peaks. But those some typical or representative the ion peak fragments ions very similar. So use that you can typically, if you are very familiar with, just to see that ion you see that is should be from which flavonoid. Yeah, immediately you can judge out because the flavonoid is so widely present in almost all the plant. Yeah, that's why I will also try to use collect some PPT, some good example to show that, so you have a very deep impression about the LCMS or GCMS, how we analyze our metabolites through the fragment ion peaks, those to track back, find our metabolite, the ion, in the mother ion. So we also call precursor ion. So you use this, after that I will get back to the first slide. So this is when I was in USA, the 2011 published plant cell paper. We try to identify what is the transporter can transport acetylcysteine from cytosol, where acetylcysteine was synthesized in the cytosolic side of ER. Yeah, and the rest in the curriculum. So those we know there is a metabolic channel, several enzymes from PAL to C4H, 4CL to CHS, then DHR. Those some enzymes are P450 enzymes. Those are anchored on the membrane. So anchored on the ER membrane. So those around those membrane form a metabolic channel. Then the continuous synthesis acetylcysteine or purocyanine. So how are those acetylcysteine transporting into the plant cells? This paper we characterized a MATE transporter, MATE, a MATE type of transporter. We show that the transporter can transport acetylcysteine metabolite, malonate, acetylcysteine glucoside malonate into the vacuole, but not acetylcysteine glucoside. So they have to be acylated modification. They subject to the acylation modification. Yeah, so the acylation malonyl group will be added to the sugar molecule of this isoflavone or acetylcysteine or flavanol. So this paper we characterize the enzyme because we find the transporter, we find the mutant have dramatically changed the metabolite acetylcysteine or flavanol profiling. That means acetylcysteine if not stored in the vacuole, it will quickly degrade, just like purocyanine, because this from Arabidopsis mutant called TT12. The TT12 is the low side, encoding a MATE transporter. That time they call the MATE1, Arabidopsis MATE1. So they already show that the MATE1 transporter, if missing this transporter, the seeds, Arabidopsis seeds, brown seeds will be transparent, with no color, transparent. Just because in any endothelium cell, there should be a purocyanidin accumulated. Those purocyanidin accumulation finally will be oxidized, generate the color, browning, the complex that mixed with the cell wall. However, this mutant in the mutation of MATE causes the failure of the purocyanidin synthesis from ER to transport into the vacuole and cause quick degradation. Therefore, their seeds is transparent. So we also find the metacarpal truncatua, one of the model legume plant. People study the forage in USA. The forage is very important. They study forage. They set up metacarpal truncatua as model plant to work on this. So we found also MATE1 transporter. At the beginning, we don't know. We just try to screen any the seeds that have defects in the fluorescence. We find also the MATE1. And we find that that's actually adopts the TT12, the homolog. Or we also can call it outlog, because each is gene exactly matched to their counterpart in the adopsis. So this is the mutation. This gene we cloned, then expressed in yeast. We try to do the transporter activity assay, but we did clone the TT12, and we do activity assay. We also show the very less sensitivity. However, if we use a substrate that was glycosylated and was added sugar, the catechin or the catechin as sugar becomes glucoside, then there's some transporter activity, but not high. Then glucoside is okay, but at a different position, they show the different activity. That means that the transporter need to recognize the specific substrate to transport those into the vacuole. So they are specific. So in this paper, we have 2009 plant cell paper, we showed the TG12 function. But in this paper, we showed another transporter can transport as a signin. They're not fluorescent. They cannot explain some, they don't show some the fluorescent phenotype. They don't transport as signin. So in this, we show that if we use the glucoside, glycosyl transferase, we generate acetylene glucoside. We couldn't get very good transporter activity assay using the yeast expression system. We isolate the vacuole, then put the acetylene and substrate. So we should be able to see some acetylene with accumulated in vacuole. Then the vacuole should be colorful, but not. Then we use some malonyl transferase. The malonyl transferase, they can transport the malonyl group to the sugar. To this sugar moiety. This is the group of sugar moiety in 6' primer position. They added this. After added this, then we can easily see we purify this product and to the yeast vacuole transport assay, and we can easily see, okay, this acetylene will accumulate in the vacuole. That means our transporter prefer to transport this, malonyl, the acetylene glucoside. And similar thing we found also in the flavonoid and also isoflavone. The flavonoid isoflavone, one is the 7% glucose, 7 position, common 7 position, acetylene is the 5 position. And in this PPT, I just want to, I couldn't see that. Just want to see that we also use the enzyme and use the LC-MS to identify the products. How to identify products? This is the enzyme reaction. If we use Utialia 7 glucoside, Utialia is only 7 glucoside. Usually, it's in 7 position, in carbon 7 position with glycoside facility. But also, delphinidine and this is the fragiline and this is the narrogine. All those. The question also have 7 position with the glucoside. So if we add the malonyl CoA, if we use malonyl CoA, we will quickly transfer the malonyl CoA to this luteal 7 glucoside and generate the luteal 7 glucoside malonate. So how to confirm this? Then we use the GCMS, all of those others. We use the GCMS to show that if we add monomial coil. You see this molecular ion. Yeah, this metabolite generates the ion, the charge here, and then compared with this one. Yeah, 287. 287 is typical flavanol, the broken down, the fragment. And this one, 441, I compared to the the 471, compared to the 53, they have 86 difference. The 86 is exactly the monomial molecule. So that means we use the enzyme to confirm they add this enzyme, the enzyme added a monomial molecule to this position. Yeah, this is the LCMS. We confirm the metabolite, the new products generated. So the same strategy was used for recent years. I think later on I will show that several Nature Science paper finished by the Stanford University. One professor worked on the alkaline, like tropium alkaline, alkaloids, and also sodium, this Jonas Center, the Anna Osborn, the Professor Anna Osborn worked on and published in Science paper. I will show that they use extensive those experiments, enzyme expression, then and feeding substrate, they generate new products, and to show that metabolite pathway is going on, then which enzyme is exactly responsible for generation of those extracted products. So we generated this nitrogen, our luteolin, our should be genistein. This is farmlanine. This is farmlanine as a flavor. We already can generate that. That means this enzyme specifically work on the cementization. Yeah, have a stereo specific selection, substrate selection. So we use this product, then we can perform this is the demonstration of molecular structure from glucose, from their aglycol, then generate the glucoside. Glucoside further in the 6' prime position with the methylated generated products, which can be used as substrate to be transported into the vacuum. So we demonstrate the mechanism of this. So this just is the metabolite. This is HPLC. We don't use that. This is LCMS to do the metabolite profiling. To use HPLC, we can easily see some major components of flavonoid. Use LCMS, we can identify some compound that metabolite that use HPLC, we couldn't reach those goals. So this we can make the complementation together to demonstrate some major component. There are also other components. So let's go back to the slides. So this just shows still we can calculate from the iron fragment, their substrate and their products that compared with this. Those iron peak, we know what's the modification happen or take place catalyzed by the enzyme. So I think that actually this morning, Dr. Werner already mentioned about the data analysis. So I think not always good to re-emphasize or from another angles to explain those data processing outcome. So generally, if we got lot of data, the mass spectral ion, total ion peaks, how we select those ions, how we identify each the peak in what. So use those. There are some if you only use the personal checking. I think sometimes the personal checking, one-by-one checking is very necessary. in many mass spec lab or the metabolomics lab, they always have some person to check those pretty often. But you cannot check one by one, exactly from beginning to the end. Those 1,100 of compounds, if you check those, that's just too much. At least at the beginning, you need to do that. But later on, most of them use those different kind of software, use those machine-like pipeline to quickly go through those metabolites. At least they can quickly give us some possible for metabolite prediction. Yeah, also the compound or annotation. So we can quickly check if this is true or not. If not true, then we use other way to further verify. Then we check it one by one. So I mean use the software and also use those kind of program to confirm. Yeah, general process just like Professor Winner already said we need a normalization of those, remove the noise, use the baseline, or we use and then keep identification to the corresponding metabolite. Actually, those is just one word, but behind this word, this extensive work is going on. And also metabolite annotation, most important, or most often what we see, is a new metabolite or unknown metabolite. This is quite common, right? And also, finally, even those peak we already corresponding to each metabolite, but for the accuracy evaluation, we need to confirm. And usually for this PCA, pre-simple component analysis, is very common for the analysis. But this usually just try to confirm each repeat. You have 5 biological repeats, and this one, another sample have 5 biological repeats. In the same machine, you run all those 10 sample or even 15 or 20 sample repeat. After that, each sample use the metabolite, all the peak ion, or if you use the peak ion molecule to classify those metabolites. Eventually, you find each represent one type. But using a similar biological sample, you can just use the ion peak, the species, to do the principal component analysis. You will find this sample, the class together, this 5 class together, this another together. The variation very small. That's very good representation. Yeah, that means that's how the principal component analysis actually do the comparison to make sure your analysis is reproductive. If 5 sample, 5 repeats, all this class together, another sample, 5 sample, all the class together. Even those metabolites put together is very similar. However, always have some difference, right? And also have some quantitative difference. Those makes the difference. So it shows the cluster green, the component PC analysis shows it. And also use that later on, you need to scale up the GC or LCMS detections to make a pipeline for more samples. That's not only for the mass lab to do the more service, but also for your own, you want to process more other samples. If only you already set up some the method, that will be very good. Just like this morning, we told several labs already set up a global service for the lipidomics analysis. One of the center is the Texas Lipidomics Center. This is very pioneer one, very early. I think the first one to do the global service. There's so many people from Europe, from China, or from US is mainland to send sample to the. So we just set up the pipeline, quickly do the reproducible mass analysis, then quickly use the machine automatically generated the Excel sheet for each component, each sample. So many samples. So their technique, this morning I showed one slide, the Mary. Yeah, she already working there for more than 10 years. No, more than 10 years. She's very good, very familiar with willing to do this kind of boring. If you repeat it many times, you will feel boring. It's always repeat those things. You repeatedly check what's this sample. If I repeat, is doing very well, or is there any problem? You go back to the machine to check those. To probably need to repeat another sample to make sure all those meet the t-test or q-test analysis. This is all been required. So this is generally for the metabolite lab they need to do. So those, this again, this is very, I just found this PPT, this one actually very good. Then for you to show that many need derivation, right? Derivation is very common. Even for LCMS, some of them need to do the derivation, just to try to give some detection chrome or other way for them to detect. Other ways, some metabolites, you only depend on the mass. Probably not enough. For example, in flavonoid, people also use the DAD or DPA, those detector. I mean, use the double. Means one time you pass through a small chamber, we will have UV from 200 to 800 micrometer scanning. So you get the absorption. So this absorption give you one information. Then the chamber go to the mass spectra, then give you about 10 minutes later, they give you another mass spectra identification result. So you can exactly, according to this mass spectra, total ion retention time, and then can match back to the UV absorption. So this is very good for flavonoid, because different flavonoid, as thymine, flavanol, catechins, and also others have different absorption peak. So if ion peak and exactly match that, probably just like 10 minutes earlier, a pattern is same. Just to make 10 minutes difference, you will see exactly this flavonoid exactly in that part of flavanol absorption in 350 around. So, but here we can see the 287, the typical flavanol ion peak fragments there. So we just make us very confident for our metabolite identification. So those derivation, like we commonly said, the trimethylation, trimethyl that's what we used for cardiovascular analysis. For wax analysis, you need to use these two chemicals mixed together, then reacted with chloroform with a leaf or some stem. Just leaves there, like less than 1 minute, so you can dry after do the reaction. And methyl mediation, just like to analyze fatty acid. We do methylation. Methylation actually called they call methyl O methylation because they are O position and maxillate. The maxillation is in the O position. So that's a very common. Then you go to generate those data. This is some common or overlapping the metabolite can be applied to GCMS or LCMS. I found this very useful. Anyway, those later you will get this PPT to recall all those lecture. So this is SBIMI. Then in last in the morning we talk about it. And the mechanisms for the headspace SBIMI headspace solid the phase extraction the SBIMI. So the mechanism just use fiber, they very easily can attract because those in the air or in headspace, those air, the molecule, the molecule, you know, move each other like a finger movement, right? But suddenly, if you close to the fiber, they will be attracted because the affinity is very good. So it will be attracted, then accumulation, then reach to a kind of a concentration. So if you only take the air and do injection, you signal weak because the concentration is too low. However, use this, not only dramatically enhance, 100-fold enhance the signal, but also the purity is good. Yeah, this is the SBIMI. And also there are other, for the fiber coding method is different, generate diverse type of fiber. Yeah, for different purpose. This is SBIMI, how they use a different variety of SBIMI matched with different other way to analyze it. Definitely you can see the SBIMI not only for headspace, but also for solution. So it means that if you have a solution extract, but the concentration very low, or you want to analyze a specific type of metabolite, then you can choose different coding the fiber to enrich that type of metabolite. Yeah, then those also can be injected into the LCMS. This is GCMS, this is LCMS. Have different injection weight. This is different. That's one injection is very convenient because but the LCMS have different need a special equipment to do the injection. But this is really great to analyze specifically some compound and enrich. Yeah, many often the LCMS analysis actually sometimes is very good, right? But when we talk about this, we only talking about their goodness, but we never talk about so many failures. We couldn't detect just see the mesh. So this is actually common. You have to realize this. So that's why the sampling or pre-treatment sometimes very important. Like use this one, use different fiber or different property fiber, you can really enrich your result, simplify the result. You can get much stronger signal, and stronger signal, high concentration signal can get the very good the fragmentation, and then much better the mass spectrometry analysis performance. Oh yes, this I put here. Yeah, in previous class I tried to say that if we analyze the lipid, from this lipid, there is some protocol like extraction method for adults or other leaves or plant, extraction method for the seeds or lipid of some animal tissue or yeast tissue for lipid profiling. And use those, you also can do the Q test to make sure those data are very reliable and can remove some unusable data. Like repeatment. If long, as long as you have 3, very good, that's enough, right? So those are the website or link I also listed. Those PPT, you should be able to see that. I will not touch it later. As I said, usually repeat expression, people will add some antioxidant compound. So overall, the procedure is raw GC or LCMS data, then you denoising or baseline correction, then do the alignment with the standards, which at more as you as more compound as you have as the standard, but this also limited in terms at all between the different sample. Some people just try to quickly compare the total iron, the spectra with the different sample. So we can quickly look at this sample has much more and this sample has a different. Or some part is enriched in this sample and some part of iron peak enriched in another sample. It basically came from the root flower or some tissue or even different plant. You can find the, because you using the same protocol to run those. Basically, it should be very similar. However, 一旦将其是 using a same molecule weight, it doesn't mean you have same molecular plant, molecule or chemical structure. Definitely, there are a lot of variety. Iron peak peaking in that case, and I told if you're interested in this, that what kind of metabolite, you can directly select your molecular weights in common. Then you just enter into to extract the interesting iron peak and do further analysis. Yeah, usually the good mass, the mass tool, like a triple mass or triple quadruple mass, you select the first mass, use that mother or precursor iron, then you will see weak signal because also go to the iron detector. So you will weakly see some peak there, but the peak very weak. Then you see that this molecule should have usually in mass spec. Later I will give you some database. You find this compound should have some iron fragment. Then you pick up this iron fragment. You know the molecular weight is what. Then enter into the second mass 2, the iron peak number. Then you will find okay, this peak is strong, signal very strong. But the earlier precursor peak becomes smaller. That's right. That's reasonable. If you insert the quadruple, insert MS, you find another the daughter or product peak. Then you confirm in that peak you also can see the 3 definitely this compound is confirmed. I mean precursor peak and different degree of fragment or products peak, iron peak. You can see in same mass reactor. That means great. The compound is confirmed. About why the show up the fragment like that, you can go to the database. They will explain why this compound is broken from this position, from this bond, chemical bond broken, and then their second MS broken down in another. So this is exactly, they always have a very trustable rule to have chemistry. That's how chemistry is reliable. That's why even biology couldn't do the industry very well, but chemical, chemistry always. So all those, then finally, this set for chemical, for the compound peak identification. Then we can do the other, the merging peak, compare with your standard, or you using different sample in parallel analysis. You can merge together to see the peak change. This go high, or this going down, or other. So the final give you the data matches. And this is one way to see how mass spectrometer can do the quantification, because this is very reliable. At the beginning, when I go with study with those royalty in Kansas state, it is only December. We do a lot of those normalization. We do those calibration. We purchase the standard like PC or TE. We know the molecule there. The fatty acid chain is exactly with that one. Then we do the different amount. We do different amounts, try to see the foundation, or mix some together, mix different PC products together. Just like this, you will see the same compound after the methylation. This is I think this is the derivatization. You can see cyclopatry, the same stem, the methyl silicon. Yeah, this is the derivatization product. So for the amine, the hexane is 6, carbon 6 hydrocarbon, and the octal is what? Octal is 8, right? Octamer is 8. Then decamer is 10. Then this one, the docan is 12, right? So 6, 8, 10, and 12. Exactly two different. So the pattern very similar, very uniform. So you can say mass actually do the quantification. And this one, then also you can use some those type of molecule internal standard to calibrate to do the quantification and the molecular, the metabolite identification. This is one of the interface for the sample. When you click this one, immediately show up the mass structure. You can find the bottom should be MZ, the charge ratio, and this many mass spectral database, not only GC, MS, also on MR database established by some government and private company or instrument provider like that. Every LC-MS provider usually generates their software to analyze. They have to, otherwise the machine just like blank. This is the integrated part of the LC-MS. So I think this one is like one of the interface for this is for GC analyzer is also a company that generates the software to analyze those. And this is another company, X3MS, the web link is here. We also generated the LCMS for on-target metabolite, why it can be used. So their procedure also very simple. You just take out what you already got, the raw mass spectral data, and put into this online. You probably need to pay some fee to order the service, then just put the data info, then we will analyze it. We will analyze it, give you the result like this. And you can check one by one. They also have database for you to check ion each peak by each peak. So those mass spectra from mass 1, mass 2, you tag the mass have 2, or the quarter of mass have 3, you can do those analysis one by one. As long as you analyze some of them, feel very confident, that's great. Then you can trust the rest of them. Right, that's how the pipeline will work. The company survive just because of the product reliability, reproducible, right? Reproducibility. So as long as they confirm some of the randomly confirm some of the good, you should be able to trust those results which you log down with just the raw mass state, you get the final, the quantified and also qualified, I mean the molecular formula and the structure of the DNA. This is another metabolomics. This is very powerful, this one. They continue upgrade from version 3 to version 5 to recently come out the version 6 one. You see GCMS, MS, NMR, also spectral beam, the metabolite concentration of those, the pipeline got very convenient from this. The sum of the three, this is the metabolite analyst, So after the data come out, you can really use the metabolite to construct in your plan or in your sample. What's those metabolites have a correlation with? You can use the PEGT, you can use the other software, other program online. PEGT is online free, and also other data. I think I will leave some. You can construct the pathway. And if you have know some enzyme, you actually only use metabolite. Basically, you can do this. And also use those to do the metabolite correlation. For example, this program is used pretty often. As far as I know, used a lot. The mapping or integrated check, data filtering, the missing value or normalization, peak alignment, the parameter optimization, and function analysis, step by step, the finished data, and finally you got for the functional analysis and make it bigger or making the other metabolite, the pathway. This is another data show. Actually, like here, they just show the pathway using their code. Each metabolite, each company, of course, in this point, those company, you can see they just try to save their time or others, but their code is only shared by their own company as software, not shared by others. So finally, you have to transform, convert into the public CIS, the CIS standard, those chemicals. CIS is very important. Every compound has their name, right? Unique name. Those are unique. So you have to convert into those metabolites to do the uniform analysis can be applied by to other software analysis. This is one of the figure we eventually lie out. Those is after conversion. You can see very beautiful the data to show the metabolite. Glycolysis, for example, glycogeneration is in human body. After we eat sugar, broken down, then generates the fat stored in our body. That's a whole thing. This is some metabolite. Finally, makes this figure, makes this beautiful thing. This is another company. And this company, this morning, Dr. Wayne already told about the GitHub is a very famous company, have not already established for a long time. They recently come out with those metabolite analysis also used for mass spectrum. One of the important database used by many mass spectral company based on many companies, based on those, just because this is the United States American state government set up, just like NCBI. I guess everybody knows the NCBI, right? You search literature, NCBI, it's National Institute of National Center of Bioinformatics. This NIST math, if you study this, you also meet this. The database, very good library of different resources. There are a bunch of people just try to enhance to work on this. The government supported. From this point, you can see the USA really some government branch just try to support global civilization, global the advance in science or in industry and other. This shows a lot of different type of. And this is the one of page which you can see. This metabolite. When you try to find out what my metabolite will generate, what kind of ion peak, search this, they will give you all the information. Then you use those precursor ion and also the fragmented ion peak. And all those number of data, molecular weight and charge, how you break it down, you can find it from here. So their number actually, they continue to increase their library, the capacity. You see from 2014, 17, and 20 to 23, the compound dramatically increased. This just, in same color, that means in the same branch. Some of them only work on the mass spectral tandem mass, some of them work on the orbi trap. It's really huge, the data. I think this information very important. They have website, so you can search. You can do the mass spectral. This is one of library. You can tandem mass spectral library. You have website of from this here. You can from this window you can go to the other related the branches of this. And this really becomes this is some useful link also, and it's not only for the lipid but also in other metabolite, the mass spectral, the database. You can search those. Sometimes you couldn't immediately find in the. Most of those you can get in AST, but some rare compound or some not common, you probably can search get from some of those. This is another way to show that how the processing. But overall, the processing I think is become gradually become simpler. The general procedure for plant sample, mass spectral data, I think it should be become simpler. When you get ready to do that, you already have so many assistants. So many assistants to finish the work. Not difficult at all. Not like over generation. You get into this. Not so many resources you can use to interpret the data. So our the scale very small. At the beginning we mainly study single unit. Look at today, the metabolite, the science or nature paper, just one time. One time we can identify more than 20 enzymes. One paper, incredible. Then demonstrate illustration the pathway, really incredible. But anyway, the later on we got that. This is another PPT. You can later you can review this. Some of them like this is data processing, data pre-processing. Use those the XZMS and then MZ gene or the meta-align or other metabolic analysis, or use the database NIST or others to do that. The artificial intelligence this morning, Dr. Winterallen used some artificial. The young generation should be should be able to catch up. We for those very fast developed AI, probably like a half month we'll have a new version to generate like ChatGPT or OneAI or the Google, ChatGPT. Every company, Facebook developed their own artificial platform. So I will just use, some QTL, the application of MS metabolite profiling used for the QTL, MQTL, metabolite QTL. Metabolite is, we know the many enzymes involved. So many enzymes means many genes. Multiple genes with those traits should be quantitative trait, right? That should be used the QTL, quantitatively trait flow cycle, right? Yeah, that's how the QTL comes. So for those quantitative traits, we have to use the QTL population mapping. You test all those population, 100 or 200 those sample analysis. The good thing for this, you don't need to focus on all the metabolites. You only mapping, you only see the sample, the difference between the sample. Your molecular marker, because you already doing mapping, you know those samples have a marker inside those plants. And only check those plants have what kind of difference in metabolite compared with others. If your markers segregated with some metabolite peak, you only want to see the peak. If the peak is reproducible and always co-segregated with those peaks, then you focus on to identify what kind of peak are those. Then this will be easier. Right? After those, you can do the further find out what the marker can, how this marker can address the metabolite difference. That's how the MQTL is working. Yeah. Yeah, this is the minutes ago I already explained to you. We actually try to use the enzyme, the activity assay to identify the enzyme activity. And similar strategy can be used for the larger multiple step metabolic pathway demonstration. This will just try to identify some transgenic plant. In top, we only express the dust, empty vector, or dust control. At the bottom, the HPLC, which expresses the viral transcription factor, which targets on the flavonoid pathway, can enhance the protein biosynthesis. Indeed, when you overexpress this transcription factor, the active protein biosynthesis in root, you can see the new peak come out compared with the control. This is the same transgenic every root. However, when you overexpress this enzyme compared with gas, you should have no any influence. Compared with as a control, you can see the dramatic difference. Indeed, when you do the mass L04 mass analysis, you want to see the more difference. Use HPLC, I just want to confirm my strategic plant give difference. And those differences should be what I expected, because the major difference is there to confirm the gene function. But then you want to see if there are other minor or other change that our HPLC couldn't detect. Then we use the LCMS, we compare the two samples, MT-PAR overexpression and gas control. Then we actually see a lot of difference. Very interesting difference is isoflavone actually decreased. When you increase prosanidin, there's the metabolic flux actually flow to the prosanidin by synthesis. Then in the legume plant, in the legume root also have isoflavone. The isoflavone reduced. That means that the enzyme actually, the gene transferrin factor actually can redirect the metabolic flux into the prosanidin by synthesis or astaxanthin by synthesis and inhibit the isoflavone by synthesis. So that's this paper, published in Plant Biotechnology Journal, demonstrates how this MT-PAR actually can enhance the legume prosanidin by synthesis to read. The day before yesterday, I told you that in USA we have this research project try to manipulate the prosanidin production in alfalfa. This very high quality legume plant. Legume and the forage can enhance the cow, the milk production, and also enhance the milk and protein level. The milk protein level is very important. That you have milk, the content, protein content less than 3%. That's the bad milk. About 3% is the minimum requirement. In China, we have lower, I think, but in the US, at least 5% the protein. So those depend on the high quality of forage. Of the alfalfa, so we can increase, not only stimulate the cow produce more milk in amount, but also enhance the protein level. But if produce, overexpress the MT-PAR and can enhance the prosanidin by synthesis, also can cause those cow or cattle or sheep, those animals have a two stomach. They have less pasture, lethal pasture death. So this transgenic plant has been licensed to a company. So I think they earn a lot of money. I work on this project. We just got a very small, because most of this gene was patented in US patent. So they already have a big application in licensed in Argentina and I think in New Jersey, New Zealand, and also in Japan. Yeah, Japan wants this transgenic alfalfa to grow, because transgenic alfalfa mainly cow or cattle is rather than human don't, so they don't care about it. They also have a higher protein and increase the meat yield and increase the forage utilization, means increase the milk protein productivity. So all of those makes the products transgenic. That's why one of my paper published in the first we published in PNS, MD-PAR, this gene we first identified the gene function. Then we do the further collaboration characterize the transgenic alfalfa, how they change the metabolite if we really produce the enzyme to reduce those muscle production in first stomach of a cow or cattle. So if only the muscle production decrease, then we'll reduce the lethal pasture gas. Yeah, they will not blow off. Like the methane production in field in the cow or in farmer is pretty high. Actually, this is one more major reason cause the global warming. We don't know. But anyway, cows every year release a lot of methane. So those methane come from forage. They digest first, the high protein will generate the methane. But if we slow down and go to the next stomach, those protein will be redigested by the cow and generate the protein milk. Okay? But first one, in first stomach, they only generate the methane. Yes, that's why we do this transgenic alfalfa test the function. Now this is the pathway which I should, yeah. You know, from phenylalanine to the commercial coil to malonyl coil, then with the CHS to all down the S-cyanine, they use pink, and also pyrosanidine is kind of brown, and also another color is for the S-flavin generation, and also flavanol here. Yeah. All of those actually need accumulated in vacuum. And we already identified those weight-1 transport anti-tracking glucoside into vacuum by weight-1 or TG12, the hormone. And then another weight-2 our EBC transporter transport S-cyanine into vacuum. And this one, S-flavin, we haven't done, but we all paper haven't published. But we did identify some. All those, the downstream pathway are regulated by MDW complex. Means those two are new transcription factor, MD-PAR, and the MT-LAP1, the medium type 1 new transcription factor, and also MIB14. And WD14 is W40, the type transcription factor, BHLH in TD8. They form a ternary complex. The complex together control the flannel biosynthesis, control the S-cyanine, mainly control S-cyanine and the pyrosanidine production. This mechanism also true in tea plant to control catechin accumulation. Regulate the catechin biosynthesis in them, ANR or let's say ANR or ANS or UDT glucoside transferase. So those complex from a mutation, from a genetic already confirmed, your mutation of TT8 or your mutation of WD40, either one mutation will cause seeds, coat, color, loss, become transparent. Not only in medical conjecture, but also in autopsies and also in soil. Yeah, those we already confirmed. So actually those complex also regulate the root hair happening and also leaf trichome. In autopsy, leaf trichome, the density of their biogenesis or development regulated by complex. But the complex only difference is the MIB transcription factor is changing. But other WD40, no change. In autopsy, the TTG1, in T plant also we call TTG1. They control why called TTG1. G is globular. Globular means no hair, right? The globular. Yeah. So those TT means transparent test stack. Those TT8 sometimes change to the EGL or the GL. So the complex mainly controlled by the transferrin factor. This is we can go through this science paper released in 2003. So we can have a very good impression, see how this metabolite can be used for identify the enzyme activity. Those are similar, much more simpler than the metabolite profiling. Metabolite is very complicated, but as I said, you don't need to worry about it. I guess in the future, there's so many software and the AI will facilitate, help you to get the very good metabolite data interpretation. So this is the Joint Center science paper. We demonstrate in this plant. There are two saponin. The two saponin we call the QS7 and QS21. The two saponins actually are normally used in the vaccine solvent. Normally, we know some vaccine we use aluminum hydroxy aluminum is like gel to make the virus inactive. Anyway, in vaccine, those we call the adjuvant is very, very important. Can enhance some vaccine, induce strong antibody and help T cell response. So those two saponin actually used in different type of medicine as adjuvant. So this, therefore, the scientists study in this plant the QS7 and QS12, how their pathway is finished, how their synthesis. Their demonstration, they try to produce, facilitate high productive or high yield those tree selection, so that to produce those compounds, because those compounds are very expensive. You just need a small amount of in each vial to as a vaccine. So initially, this is the abstract. This paper we identified three enzymes. This is the most major three enzymes for QS7 and another for the QS12 by synthesis. So this paper actually from study focus on this tree, and they finish the genome assembly. Because to demonstrate a pathway in an unknown plant, you have to finish the genome sequence. That's how big paper usually attached with the genome. Right now, the genome sequence for a plant is very cheap, not like many years ago, very expensive. But now it's cheap, assembly. Even to a transcriptome, you can quickly get some Beijing enzyme. During that time, you can do the genome sequence to assembly it. And get the genome sequence is actually very important. The Indian scientists also recently published the Asamic type of tea. Genome sequence. That's very necessary. You can really understand what's the plant, what's the tea plant in your country, and the soil you produce so many high quality tea you everyday drink, you still don't know your plant. That's not so they either the genome sequence on this plant, then use the genome sequence assembly the protein or enzyme, then do the the Q7 to S12, the bisensitive pathway demonstration, those compounds that can use for the vaccine. They're very useful and very valuable. Just quickly. This the first way. The first we show that structure, the difference, digestive structure of what they have glucose, they have a Reynolds modification, they have acetyl modification in the fructose and Reynolds so completed xylose and anti. Then I have the glycol, all those the compound in the fibrin saponin. And we use the MS to confirm those metabolites, the structure very well. Then those red already. Yeah, you said you try to confirm those modification. And in those hydroxy group, you will have the glycosylation modification. This the purified enzyme. The paper actually, the website I showed you, you can read the paper. The long, but very exciting. The step by step. The first they demonstrate this step from QA to QA monoglucoside, then diglucoside. Yeah, it should be diglucoside. And then digranuloside. First is the galacoside, then adds the granulocyte. Here we have trigranulocyte and tri-xylose. This is they finish the genome sequence assembled the genome and they identify the full protein sequence, then gets the sequence expression, and just like previous what they found in aldopsis, and they found those glycosylate transferase and also P415. They have showed the cluster. Yeah, show the cluster in one chromosome. They're very close. Of course, those cluster not included. There's some one over the P415. Many of the others are glycosyltransferase. And beta NS or other OSC, the things not here in different chromosomes. But still shows those amplification of pattern duplication in genome level should be responsible for the high level accumulation of extensively modified saponin. Yeah, this I think that this PPD should tell you the pattern duplication of the glycosyltransferase saponin responsible for so many extensively modified saponin, like tri-xylose, again at the fructose, right? Another sugar, fructose. Then this tri-valence and fructose ranose. Then and fructose ranose. This is called banana. Anyway, the tri means 3 xylose sugar added. Then the F is fructose, right? R is another ranose. Xylem is another xylose added. That's why the sugar chain is very long. All those have different type of glycosyltransferase. This is the cereal. Yeah, from the pathway, from beta AS, is one of major branch enzyme synthesis, the beta amylin. Beta amylin is the basic backbone for supporting biosynthesis. Then first, the cyclase. This CYP 716824 should be bound by OCS. Cyclase then form oleinic acid. Then continue form another cycle. Hydroxylation, P450 consecutive hydroxylation, but it generates the quinic acid, then continue with those glycosyl transferase generate the step-by-step successive glycosylation, then generate, but still not to the Q7, QS7 or QS21 yet. Yeah, this is kind of difficult, but still we continue to identify other QA derivatives through the glycosylation reaction that generate so many. They also have a USD. Yeah, some of either this one is another Reynolds and then glucose, then generates the QS7. They just reach the first goal to demonstrate how Q7 generates step circulation. Generates the backbone, the quinic acid, then based on quinic acid, the successive glycosylation in different backbone or in the sugar moiety and different hydroxylation and the sugar like this in here and also from different position and another sugar. A different type of sugar. This is the try to demonstrate how we generate the QS12, I think. Also, this is this is another paper actually, also from cocaine. You know cocaine, right? This is like toxin, like drugs, right? This is drug, but well, this is another Nature Communication paper. Also use the same way to demonstrate the PK. The cocaine actually is through this pathway, polypeptide synthesis, then continue to the oxidation. Then P450 and hydroxylation. As long as the hydroxy group, then have a monomial MT. The methylation, M4 methylation, then continue got the cocaine. The final step is this cocaine. A different way to generate that. The very interesting part is that this part, they show that why the different plant, the phylogenetically did not correlate here. Related the compound, the plants, but produce those similar products. You see, this part is the solanus, right? Solanus plant. This is the milk pickers, different type of plant that generate the cocaine or the similar kind of protein alkaloid. So those we also call the convergent evolution. In phylogenetically related the plant, but generates the similar secondary metabolite. All of this because their genomic duplication of PKS. There are also other key in that. For example, this part is the ST, MT, methyltransferase. Usually methyltransferase usually can facilitate generate some conversion to evolution products. For example, caffeine. The caffeine also like several position methylation, NMT. Unmethylation actually unmethyltransferase, the enzyme originated from O-methyltransferase. O-methyltransferase is the only enzyme. They generate like jasmonic acid, methyljasmonic, also methyl salicylic acid. They belong to the O-methyltransferase. However, in recent years, we know that not in recent, like in 30 million years ago, not recently, 30 million years ago, the tea plant evolved so many unmethyltransferases through genome duplication. Whole genome duplication or the gene duplication generates tandem arranging NMT. The some NMT, for example, this I think this part is very interesting. But anyway, in this case also this is alkali, okay? But in tea plant or coffee, cocoa also have caffeine. But those are not related. Okay? These plants are very different from each other. The similar things are also people identify some the red pigmentation in in plant. In plant kingdom, the red pigmentation can classify into only two types. One is acetylene. Another type is right? Baiting, right? The baiting, right? They belong to the different another group like baiting, B-A-T.
IT.
The baiting, yeah. Yeah, this is another red color, right? They also derived from aromatic amino acid.
Biotin.
Huh?
It's biotin.
Yeah, biotin. Yeah, they also from derived from I think from tyrosine, right?
So erythrin is the red color we see.
Acetylene derived from phenylalanine.
It's anthocyanin there.
Is
Yeah,
Red
yeah.
color.
Acetylene derived from phenylalanine. But the baiting derived from
Anthocyanin gives
tyrosine.
purple color anthocyanin.
Yeah.
Asking about the
How about the baiting? Baiting is also red color. In some
Red color.
in some tubular.
Erythrin gives the red color.
No,
Erythrin.
sorry to say, red color is not given by a single even metabolites. It's the pH that matters. So many metabolites influence, give the red color.
Yeah.
We can't say that it is only anthocyanin. Some other metabolites also give the red color,
Yeah.
and that depends on the pH of the vacuole and some cytoplasm.
Anthocyanin gives the purple color.
No, these statements are not accurate actually. Colors in part by some other issues actually. Acidic medium, basic medium, many other things. Yeah, please carry on.
Yeah, actually, that's very interesting to just use metabolite profiling of very different plants. For example, that one paper published in Nature Biology Chemistry, Nature Biology Chemistry, Nature Chemistry Biology, or Nature Biology Chemistry. But anyway, this journal just studied why the baiting only present in several very different plants. They also call it convergent evolution. Suddenly disappear. where somebody appeared in this group of plant, but other plant don't have. And you don't know the probably in my pronunciation. Please check the, tuber. Sometimes we eat like a, they have a red color.
Yeah, anti-tannin tablets. They have some infection,
No.
sometimes
No,
chilling stress also.
not as many. You breathing. EAT. Could you please check the website?
What's the spelling?
Huh?
What is the spelling?
DAIT. If you check the different type of the pigment, two major type of pigments
PAT?
in plants.
PAT?
Yeah, PAIT. I should check the. Yeah, that story very, very interesting. Actually explained in nature, in our plants, there are two types of those different molecules.
Retaining,
Yeah, breathing.
retaining.
You find it?
But he is saying by PAI, na? Spelling is?
PA.
Just tell me the spelling. PAI.
BATAININ.
P or T?
P.
P, no? It's
P
okay.
for
Yeah, A, B, C, D,
pick up.
P.
Pick up. Pick up. Pick up.
Beetroot is not a fruit, it is a good food.
It's covered by the red.
Beetroot.
Vitamin.
Could you please check? Yeah, pigment. Yeah, what the soluble pigment that you plant. Yeah.
So this one, A,
Petalin.
Yeah, between. Yeah, between. Yeah, between. Between is derived from.
It's a water soluble
Yeah,
red pigment
yeah.
that comes from the red beans.
Yeah. Those are not arsenic. Those are not arsenic. This is also derived from the tyrosine. Tyrosine and the phenylalanine all from same pathway. Same pathway, skinnic pathway. Sneaky acid pathway. They generate the silic acid. They also generate the three aromatic amino acids, right? Tyrosine, tryptophan, and phenylalanine. Yeah, phenylalanine give arsenic. However, tyrosine also give the pigmentation, but not arsenic. If you check the tyrosine, then the scientists study why those plants don't use phenylalanine, but use tyrosine to generate the pigments. Those pigments, one fruit, the red is very colorful, like red. I think, what's fruit called? You don't know, you should have it, yeah. Oh.
do you have phone?
Huh?
Do you have phone? Your phone?
Yes.
Think different. And even one more interesting thing I would like to tell you, if you go China, your name will be the change. They will give you a different name. And you will be recognized by that name, Sang Chan Chan Dodo. Here, that was written on your passport also. There. Whatever you book, whenever, they will pronounce you with that name because in that language our name are not exist actually. So that's why they are using the different names. So that's a misunderstanding here. Whatever the name sir wants to explain us, that is local of the China, but here the name of that item, that plant material is different.
No, actually, this is tropic food.
Tropical food.
Yeah, tropic food.
And you go to the
No, it's no network.
go first, go ahead. Go there first. Sir, yesterday why we are blessed with as compared to the Chinese. Reason is that if you purchase a land here, it will be forever with you. You and some grandson, ancestor property. But in China, it is only for the 70 years.
Yeah, it is a lease.
Lease. And after 70 years, there is a lot of the taxes you have to pay, and that you have to leave as soon as possible. You will be enforced from that place. The another important point is every youth generation has a lot of the pressure to get money as soon as possible to purchase the home. When they purchase the home, only after that, some girl ready to get the marry with them.
Dragon fruit.
This is very famous. So now we try to understand every generation from the early childhood they start to get the money, deposit the money, and remember the most expensive thing in the China is to get the home actually.
Yeah.
So there is a lot of the pressure on the youth. If they want to marry, first of all, their dream to get a house actually. And it will be only for them, not for their kids. After 70 years, they need to get another house also. So that is a thing. You can think how much we are lucky, and that's why we are also very comfort. Our youth generation are very comfort. They are not ready to get the serious attention of many things, and they are not ready to get started to earn the money at the early age also. They want to enjoy from their parents' properties. And after some time, they are telling their parents that what you did for us. This is the real scene. After 30, we are telling our parents what you did for us. So try to understand, this setup is the different in other countries, and we are really much in comfortable social security and other things. I also requested, do you get the water and the food free of cost in the China? Sir told me no. Without free of cost, you can't get the food and the water. But in India, you can easily get food and the water.
Yeah, that's it.
So that's the now this dragon fruit is also cultivated in the India. It is in the market. Yeah. So please go ahead. You must stop and
See, the natural, the world, the nature can produce much diverse pigment. If you're interested, please check the dragon fruit, the pigment, not as sunny. Many people in China even confuse. I said it's not as sunny. It's different from our color or most other place, most other plants, the red or purple. The as sunny can be a purple color. Of course, because of different molecule, like delphinidine. The delphinidine, these three types of major.
It is beta cyanide.
Yeah, beta cyanide. Yeah. So those delphinidine generate the purple in acidic condition. Usually flower can be acidic. Why some plants, the flower is poisonous? Just because one thing is because of the mutation of those transferrin factor is tissue-specific expression, or just use development stage specific expression. So you can see different patterns, the spots, dots everywhere, irregularly present. And the delphinidine can respond for the purple color, the ascending. And also, another one is the stannine. Stannine glucoside is responsible for the red. But the protonidine only have one hydroxy group, ACB. You know, every flavonoid have ACB rings, 3 rings. So on the C ring, ACB. Yeah, on the B ring, there are some 3-hydroxy called delphinidine. So in catechin also, in T, catechins also have 3 rings. You get catechin, EGCT, all those. That's why catechins have so many hydroxy groups that they have very strong antioxidant activity. The number of hydroxy group present in the molecule represented the antioxidant activity level stronger. The more hydroxy group, the stronger the antioxidant activity. But anyway, assigning and the catechins or epicatechins, those and another group of catechin actually, another group of catechins also the corresponding to assigning because they basically from similar pathway. All those assigning or prosanidine group from derived from phenylalanine. However, the betaine they derive from tyrosine. Why? The tyrosine also subject to similar D-amino acid, deaminide the group. You use the TAL, the phenylalanine amino hydrolase. Yeah? Remove the amide, NH2. Remove that to become the acid, called the cinnamic acid, right? Yeah, cinnamic acid continue become comuric acid. Comuric acid become comuric CoA, then together with the malonyl CoA form the phenyl propanol pathway. Then continue phenyl propanoid pathway leading to the lignin production. Right? Lignin and some part of UCHS diverts to the metabolite into flavonoid pathway. But HCT divert those comuric CoA and malonyl CoA divert by HCT into the lignin. Lignin, S-lignin, G-lignin, H-lignin, and cause the different types of plant. Some plant higher, and some plants like grass plant. They don't have a very strong. Different wood have different type of S-lignin or G-lignin or H-lignin. The lignin type of polymer different. So ascending also different. Ascending cause in different plants have different the hydroxy group. That's why their distribution also different. Like tea plants have so many catechins, but they don't have much ascending unless there's some variety. The purple leaf. Usually tea is green leaf variety. There's some purple leaf variety produce a lot of ascending. However, the catechin decreases because the pathway is competent. You produce more ascending, the catechin reduce a lot. The two pathways come competitive for the end product. So all of those they're just similar to the betaine biosynthesis from tyrosine, the amine, generate the acid. The acid continue the several steps. The paper from Nature Biological Chemistry just demonstrate why different plants they can synthesize the betaine, but not ascending. The same color but very similar biological function attract the bee to pollination or absorb the UV light for the protection of plant against the reactive oxygen production. So they from the enzyme activity assay and from gene sequence find out actually the enzyme mutation cause the activity suddenly increased. Then lead the products to the betaine biosynthesis. Yeah, that's a very usually convergent evolution. For catechin also similar to this cocaine and also same type of 什么 ILC cacoin or other type of cacoin. The PKS, PMS, this is putrescine. Putrescine is the polyamine, right? One polyamine usually the primary products then is a precursor for the secondary metabolite. So those PMT actually is the major reason, major mutation of enhanced activity or mutation because we can take different products to generate the cocaine, different type of cocaine. Caffeine also, only in plant, in sear, in some of the camellia plant, you can see high level of caffeine. But in cocoa also, the different varieties, they have some variety have a low caffeine in tea and also cocoa. Now people try to reduce the caffeine plant. That's why my lab usually in finding some wild tea relatives in Yunnan, Guizhou, and other close to the Assamic area. Yeah, and you pass by Yunnan, you will be. So we have anyway, this area is the origin of the tea plant. But now here you see finish the Assamic Indian Assamic plants, the genome sequence probably help to demonstrate the origination of different domestication and even spread to Kenya, Sri Lanka, or other plants. Right. So, but I also in their paper, because I reviewed the paper, I see very interesting result and very good contribution to the scientific research community. They found the caffeine content also different. There are some parts very high, some parts very low. They compare to Japan variety. Japan variety as caffeine decreased a lot because Japan don't like caffeine. So the domestication and the humans intended selection very important. So this is just through the different metabolite profiling. They already scan in different plants. Try to find it. Actually, they find those plants. We find those plants. But does those plants, do those plants already cover all the plants contain caffeine or caffeine similar derivatives? No. Definitely no. That's what also I know. They said that what they have done is just screening what the literature reported, then do the metabolite profiling and find out the related products, and then find out the transcriptome, the expression of P or S, the putrescine or spermine, those polyamine methyltransferase, the gene, then found that they are enriched and you have similar the function. But through the limited number of the plants from different family, they can basically reach this statement, the convergent evolution of those genes, or cause the scattered distribution of the caffeine or caffeine, caffeine is cocaine or cocaine related the metabolite in very different scattered, randomly scattered. It's just the kingdom sometimes we have mutation, the plant kingdom evolve this product. Similar like caffeine. That's I think another way of metabolite profiling together with the RNA-seq, the transcriptome can understand from plant metabolite and their corresponding enzyme responsible for the generation of this metabolite or from different substrate. We can really track back the evolution and in different plant reaction. And this is even in larger view, you put all those plants together just to check those acid, spermine, methyltransferase, and the putrid methyltransferase of the PYKS, poly catalase, and PKS, those genes. You can find out because PKS provides the basic structure for the later on. So they can find out how the evolution of a convergent evolution will happen. Oh, is it M?
It's
Yeah, I think I am.
end of slideshow.
Huh?
End of slideshow. Yeah, this was the last slide.
Yeah, can you? It will be the last slide.
Okay, yeah, no problem. And this calculus also, not be sure. This is the reaper vine. This is the tree called pattern. This is the tomato, right?
Yeah.
This is the tomato.
Yeah, tomato. Learn like
Yeah. So what do you think about this? The metabolite really can. Actually, I'm now just to try to use the metabolite. I collect different tea plant varieties, including the wild type tea plant, the big tree, or sometimes is like we call the amber tree. And some, most modern tea cultivar are shrub. You already know obvious the trunk, major trunk. They already divided the branch out into the smaller, lower the shrub. So those evolution, or human selection, what's happened for those from a big tree to a shrub. So this evolution, this is also kind of the shrub should be it should be evolved tree. However, amber we always regard it as a primitive tree, right? Yeah, primitive tree. But the shrub is better. But yeah, now currently most the modern tea cultivar. So I collected those amber tea, wild type, and also some in Yunnan, Guizhou, or Guangdong, Guangxi, there are some shrub, or half amber tree. They show some branch, the lower, as a tracing. I think like tracing species.
Yeah, trace.
Yeah.
Trace farming.
Yeah, tracing means intermediate species in evolution. After that, if they cross with, because the tea plant is an outcross plant, the very low level of self-pollination, the self-incompatible plant. So the outcross, that's why their genome the heterozygous stage is pretty high, like 11 or even 17%. So we do the first genome sequence published in PNAS in 2018. We chose one of local variety, very low heterozygosity. So the genome complexity is lower. So we did the genome assembly with the back. Construct the back clone, then do the genome sequence, then assembly the rough genome. Published the PNAS in 2011. We found they have 2 times the whole genome duplication. And we also found that there are some genes very specifically expanded. This gene family expanded. This is methyltransferase. You know, this is like an SL transferase. This SL transferase exactly explains why T plant accumulates so many soluble pyrocyanidium. You know, T plant is the exclusive plant accumulates so many soluble pyrocyanidium. Most other plants you can see this bark, leaves are polymer, pyrocyanidium polymer. All those pyrocyanidium have same taste, have astringency taste. We put it in the mouth, our tongue was, very rough. Yeah, just because they have too many hydroxy group. The hydroxy group, denatured our protein on the, our taste, the tongue protein. That's why it causes very tough. We call it astringent. Also the bitter taste. But the reason finally, we still don't understand. But one of the SL transferase we call SCPL, the gene family, is extensively expanded in T plant. Then recently we also found that this enzyme can catalyze gallic acid. And after gallic acid glycosylated, generates the beta glucan. After generating beta glucan is this gallic acid, then this is sugar. This can be an acyl donor, transfer the gallic acid to the epicatechin and catechin. The generate epicatechin, gallic epicatechin, so epicatechin. That's epicatechin couldn't be quickly divided into the polymer. So gallic acid, after glucoside generates beta glucan, then together with the epicatechin or further epicatechin generates the AGC, TCG. Yeah, those. So we found this, and we also demonstrated the catechin. Yeah, the catechin.
I think it's enough for today. So, yes, sir told about the real key metabolites which are present in the tea. And I worked on the tea and doing my PhD. So, one slide of the sir was the paper I used in my thesis also in 2013. So I saw that slide, fluoridine and the diversity of the metabolites with respect to the pattern of the hydroxy group. If you see the research that is carried out by the Professor Jain, it is really appreciable in terms of the secondary metabolite, especially the tea actually. So sir is working on the tea plants and yes, doing very good. And now we are getting the sense that what is the level of the Professor Jain research right now. So yes, it was a very elaborated lecture of the 2 hours continuously. It's very difficult to speak continuously 2 hours. And yes, sir come up with the patience and he explained in a very understanding way. Now I think all of you are able to understand what sir want to say because time's required. So thank you for today. Yes, we have this CIL visit and we have the GC and the MS. So Ms. Ritu is calling me, so I will just convey the real message within the 2 minutes. You can go in the CIL, see there is a person, technical person, he will explain either the GC or the number or both. Otherwise, we will conduct one more visit with the CIL. Second announcement is regarding the tomorrow. So tomorrow, Sakshi is here or not? So where is the Sakshi? So yes, tomorrow whoever want to go, at least they write down their name, roll number, their mobile number, and from there they come here actually. Because we need the list who would like to go this Hosaniwa border, and we would like to make the call to anyone if another one is the switch off for any inconvenience. And tomorrow the bus, as I told, the bus will come at the 10 a.m. here, and you have to start your journey at the earliest. And I will also request the person in the mess, they will make some heavy breakfast little bit, so that you take your breakfast here, and then because you take the dinner at the little bit late actually. So meantime, if you carry some snacks, so it would be good for you. And the another point is that it is around 100 kilometers, and 30 kilometers is the one way, otherwise it's the 4-lane. So when you will visit the Hussainiwala border, so please take your I card, because there is this army men, they entered in their Aadhaar number or something like that. If you have the form, then it's okay. And yes, that is a very good place. First with the Bhagat Singh and Sukhdev and the Bhatukeshwar Dutt. And yes, there is another Indian and the Pakistan border. So you enjoy there and get your whatever the time you want to spend there, then come by the bus. Meantime, at around 8 p.m. you will get the hotel where you can take the dinner, but that dinner is from your expenses actually, because that is not, we can't afford, and we can't say, give the audit how you spend money on there. But yes, in the morning you take whatever you want to take from the mess, because then they will definitely arrange for you. So so thank you, Professor Chen, for this section. We can buy down
some of my paper.
पेपर
सर उनको उनका
कन्वर्ट द मैसेज टू द रितु कि अभी हम जाने से पहले आपको सारे पेपर्स देंगे, और आपका जो पेपर है वो 50 नंबर का होगा लास्ट डे. आई विल टेक इट अमॉन्ग द 50 मार्क्स. 15 इज द ऑब्जेक्टिव एंड द 30 मार्क्स इज द शॉर्ट सब्जेक्टिव. जिसको आता है जितना लिख दीजिए, नहीं आता कोई बात नहीं. Why Ritu told me कि sir we not need to give the subjective. So I told nahi, we need to give the subjective, otherwise it will create the problem for us also. And yes, your certificate we are going to order very soon. Let's see who will get A plus, A or B. It depends on some of the factors also. So Ritu क्या करें? अजित है?